Welcome. This is Biology Form 3. I will take you through transport in plants, part 2. As usual, my name is Bellington Kamisa. We are going to look at the transport system, but in this time, we are going to look at the tissues that are involved in transport system. The success criteria, what is it that you should be able to know? Your expectation you should achieve at the end of the day. By the end of this lesson, you must be able to define the term transport system. You must describe the vascular bundle. You must conduct experiment on transport system on the phloem tissue. So basically on the investigation, which is an experiment, you are supposed to actually show whether the phloem tissue is really conducting the food organic materials. Transporting plants, in the previous sections, you read that photosynthesis uses water, carbon dioxide to make what? Glucose as the product. So therefore, for this process to take place, water must be must be transported from the soil into the roots of the plant, then rise up the stem to the leaves where it is used for photosynthesis. So generally, water must move from soil into the plant, then must rise up to the roots where now water is used as a raw material for the process of photosynthesis. In this section, you learn about transport system in plants that enable movement of water in the plant tissues. Transport, therefore, let us define that one. Transport is the process whereby substances move from one part of a plant to another, from the roots to the leaves, where the food now is being made. So these substances include water, mineral salts, and plus manufactured food. So the manufactured food is transported from the leaves to the roots, where it is now stored in tubers for consumption or for use in the next for the next time. So transport system is made up of all plant tissues that move substances from one part of the plant body to another. So therefore, the system is composed of structures which we call the conducting structures, or we call them the vascular bundles. So these vascular bundles are very, very important. The cross sections can be used to analyze those vascular tissues as we did last time. So to identify these structures, one can carry out the activities. For example, if you want to identify tissues that are involved in transport of substances in plants, you do have materials such as charts, diagrams. You may have diagrams of those sections. Maybe you can also download them from the internet and then analyze them. From that one, you can examine the charts and the diagrams of cross sections provided of xylem as well as the phloem tissue. So you can study these tissues, the xylem and the phloem tissues on the charts. So in each section, you identify the xylem and the phloem. So from the charts that you have, you can identify which one is the xylem and which one is the phloem. Then thereafter, you note how the xylem and the phloem are arranged. So here you are looking at the arrangement of the vascular bundle, as we did in the monocots as well as the dicots. It's more similar to this. So from the activity, you realize that the conducting structures in plants is composed of the xylem and the phloem tissues. So we have the xylem and the phloem tissues, which are the conducting tissues in the plants. So when found together, they form what we call the vascular band. So collectively, the xylem and the phloem makes what we call the vascular bundle. So you now learn about the structure and function of each of these tissues. And the next slide, we have to look at how the xylem looks like. What are the structural parts? What are the functions of the phloem and like? So let us start looking at the structure of the xylem and its function. What is and basically we're looking at the anatomy and the physiology of the xylem. We're looking at the parts and functions of the xylem. Now, the xylem tissue is a specialized tissue which is having modified cells to carry out two functions. So the xylem tissue has got two modified cells, and these cells perform these functions. Now it is there to transport water plus mineral salts from the soil into the plants. So the xylem can transport water plus the mineral salts to the leaves, where now the leaves can use water for actually food making process, and the mineral salts are used to make, others are used to make what? The green pigment, which is chlorophyll, to give support to the plant. So the xylem, apart from transportation, it provides support to the plant. So generally, the xylem has got lignified walls. Those walls provide protection and support to the plant. Right, now the xylem contains two types of modified cells, and let us look at these two types of modified cells that are contained in the xylem tissue. So we have the tracheids as well as the vessel elements. So there are two types of modified cells that are in the xylem tissue. We have the tracheid as well as the vessel elements. Now let us look at the tracheid now. So the tracheid are empty dead cells. So these are not living cells. The tracheid are empty and the dead cells. They are elongated, meaning that they are long and they have tapering end. Tapering end, we mean they have a pointed end. Water passes through them easily because they have no cellular contents that would otherwise cause obstruction or block the substance that is being transported. So we are saying these have no cellular contents, meaning they don't have the nucleus or the contents of the cytoplasm, the Golgi apparatus, and all others. The mitochondria are absent in this. That's why we say they don't have the cellular content. So their walls are reinforced by strengthening material known as lignin. So lignin is a tough carbohydrate which actually reinforces the walls of the plants, therefore providing support. So the walls also have tiny pores known as pits. So the tracheids have got the walls which have got tiny pores known as the pits. So tracheids are arranged end to end and also side by side. That's the arrangement. So generally the tracheid cells are arranged side by side and as well end to end to ensure transport is done. So water passes from one tracheid to another through the pits. So the pits can allow water to move from one tracheid to another. So they offer support and conduction of water as well as mineral salts. That's what the tracheid do. Let us look at the structure. The structure of the tracheid. Now the structure of the tracheid. You can see now it has a tapering end. This end here is what we call the tapering end. So the tapering end, we mean a pointed end. As you can see, this is a tapering end, meaning that this part is sharp and pointed. So these are the pits which are distributed all over to allow the conduction of water from one tracheid to another. Right, let us remember that apart from the tracheid cells, we have also the vessel elements as one of the modified cells that are contained in the xylem. So they are very long tube-like structures which are continuous from the root to the leaves of the plant. There are several vessels are found side by side. That's how they are arranged as the tracheid. The two are dead tissues and have no cell contents. These are dead tissues also. They don't have the cellular contents, the nucleus, the mitochondria, the Golgi apparatus, or the other contents. This allows water to move through them freely without any blockage, without any in fact, without any obstruction. So their walls are reinforced with lignin, which makes them strong and rigid to give support to the plants. So the plants are well supported because this lignin is now reinforcing the walls of the plants. Right here with me here we have the structure of the trachea. Of this basically is the structure of the vessel element. Now you can see it is also arranged end to end. This the ring bordered the pit. We have the lignified walls. So these walls are lignified, meaning they have lignin. This space is the lumen. Then we have bordering pits. Then we have the annular thickenings. These are the annular thickenings. Sorry. We have perforations. Sorry. We have perforations in end wall. These are the perforations. Then we have perforations end wall of the vessels. These are the perforations. So basically, this one is the vessel element. Now, we have to look at the structure of the phloem. We have now analyzed the structure of the xylem. We have said that it has two modified cells. We have the trachea. We have also the vessel element, which we have just seen in the previous slide. But here we are going to look at the structure of the phloem and its functions. Phloem transports the manufactured food from the leaves to the rest of the plant. So basically, the phloem is there to actually transport food which is made from the leaves into different parts of the plant, where now the glucose is used as a raw material for photosynthesis, for respiration. Our goal should be respiration. So phloem tissue is made up of sieve tubes and companion cells. So you can see that this phloem structure is made up of the sieve tubes as well as the companion cells. So these are the companion cells. This one is another companion cell, another companion cell. And again, we have the sieve pores. These are the sieve pores. Here we have the sieve plate. This is the sieve plate, another sieve plate. Now these are the sieve tubes. So you can see these are the sieve tubes that run along. These are the sieve tubes. Another sieve tube is there. So we have the cell wall. This cell wall is also this part. The whole of that is the cell wall. Right, so we have the sieve pores. There are pores, the opening here right away there. So we have the fine cytoplasmic filaments. So these, each one of these cytoplasmic filaments are making the sieve tubes. So right, we have the organelles which are placed against the cell wall. This one, these two are placed against the cell wall. We have these fragments which are pieces of the endoplasmic reticulum, this structure. Then coming to this end here, we have this prominent nucleus is well developed. Then we have the dense cytoplasm, active dense cytoplasm containing numerous organelles. You can see it is called a dense network cytoplasm because it contains more organelles. Like you can see the rough endoplasmic reticulum, the mitochondria, this one, and all that. So you can see that really this one is it. Now the companion cell is full of mitochondria to generate energy for active transport of the glucose. So this one is the structure of the phloem. So we have said consists of the sieve tubes as well as the companion cells. So these are the sieve tubes, and the companion cells are having the active dense cytoplasm because it is having several organelles that are performing particular functions. For example, I've said it is having the mitochondrion, which is generating energy required for uptake of active transport of glucose from one vessel to another, from one tissue to another. So here we are just describing the structure. We have the sieve tubes. So the sieve tubes are made up of cells, which we call them the sieve elements, which are arranged end to end with each other. Now the sieve elements are separated from each other by a structure which is the sieve plates. So now these sieve elements are really separated from each other by the sieve plates, which have pores or perforations in them. From sieve tubes, unlike xylem tissue, are living tissue. So this, unlike the xylem tissue, we said it's a dead cell, has got dead cells, but now this phloem is a living tissue. So they have fine cytoplasmic strands that run through from one element to another. However, the cytoplasm of the sieve elements does not have a nucleus as well as several cell organelles. The phloem also consists of these the companion cells. So each sieve element has an accompanying cell known as the companion cell. This cell has a dense cytoplasm, meaning has a prominent nucleus, and the other cell organelles like mitochondria, the Golgi apparatus, and the others, the ribosomes. So these take up the roles of those organelles that should have been in the sieve elements. Remember, sieve elements not have the organelles. Right, structural and functional differences between xylem and phloem. Now, when you look at structural differences, you are looking at how are those parts different, right? Let us start with the xylem. Let us start with the xylem versus the phloem now. So the xylem is made up of made of living tissues. So generally, the xylem is made up of dead tissues, while the phloem is made up of living tissues. So you can see now that the xylem is made up of dead tissues, while the phloem is made up of the dead tissues. That is according to structural difference. Then functional difference, we are looking at how different are they in terms of what they are doing, what they are performing, what role they are playing in plants. So we are saying the phloem transports water, mineral salts. It also supports the plants, while the phloem, it is involved in transportation of organic food substances from the leaves to all parts of the plant. So you can see that now the xylem, in terms of function, is different from the phloem. So the xylem can also support the plants, while the phloem does not provide support to the plants. Right, we have the experiment, the activity which we can conduct to prove whether the phloem really conducts glucose, conducts organic food materials. Now, what experiments do we use? We use two approaches are used. We have what we call the ringing experiment. The ringing experiment is used to prove whether the phloem conducts glucose. At the same time, others may use the aphids. The aphids are also used to analyze the contents of the phloem. But generally, in this one, we are going to focus on the ringing experiment. So this ringing experiment is used to show that substances are translocated through the phloem. Now we have the apparatus and the materials. What materials are required in this experiment? One, we need a tree or a shrub with so many branches. We need a knife, which we use it to cut the tree or shrub. Then what is the procedure? The procedure there, we are looking at the steps that are followed if we want to conduct this experiment. First of all, we have to remove completely a ring of bark with its phloem from two branches. Then the xylem tissue, which makes up the bark of the stem, is left intact. We don't tamper with the xylem. The procedure continues. Now, the setup is left and disturbed for 4 weeks, and it is shown as here. So this is the area where the ring has been removed, is cut. This is the bark, a bark intact, not tampered with. This a bark is the skin of the plant or the stem. Then this is another bark intact. So this one is taken from Excel and Succeed Biology on page 3, book 3, page 65. Now these questions are just guiding us what could be the observations that are made in the stem after 4 weeks. After you remove the ring here, what could be the observations? Then explain these observations. So these observations must be explained after the whole 4 weeks has elapsed. Now the discussions, what could be the possible results? When the ring of the bark is removed, the phloem beneath it is also removed. Now after several weeks, a swelling above the cut ring is noted. So there is a swelling which is noted above the ring. So therefore, the swelling is due to the accumulation of food substance that were being transported from the leaves but could not get across the debarked part of the stem. So as a result, there is no swelling on the lower part of the ring. So generally, because the food is failing to cross where there is a ring, that's why it starts swelling above the debarked region. Yeah, so let us see the results here. What it means is, as food is made in the leaves above the stem, then that food is transported and is failing to cross here because there is a ring. And as a result, to start piling up, start gathering, starts accumulating here. As a result, this will cause this part to be swollen. So that's why here we have swollen part of the back. This part now starts swelling up because the food is failing to cross and reach the root section here. So that's the whole essence. The diagram again has been adapted from Excel and Succeed Biology, book 3, page 66. Yeah, you have to note that this ringing procedure is sometimes employed to kill some unwanted trees before they are cut down. So those people who are working under the forest, sometimes they employ the ringing exercise to kill some of the trees that are not required. Do not use it on a tree that has an economic value to the community because that one can kill it. Sometimes some trees have got an economic value. They may produce some fruits which are sold. For example, oranges as well as the mangoes. These fruits have got some economic value to the communities. If they are cut down, obviously people will start suffering from deficiency disease. The fruits generally they provide what? They provide the vitamins which can be prevent some of the visional impairments and the other things. So that's why they need to not to be cut down. You have to discuss with your friends how the bark of the medicinal trees can be harvested without killing these important trees. So, some trees have got some medicinal values. People get medicine from trees, from the communities. That's why the ringing experiment may not be used to actually to show on these experiments to prove whether phloem translocates organic food substances. So that's why maybe one may employ the aphids. Now, aphids, how are they used? When aphids are allowed to suck the sugar from the the sugar from the back, and again, once it is sucking, instantly the styrate now is cut off. That mouth of the aphid is immediately cut off, and that one is now dissected. The styrate is now dissected to actually analyze the contents under the microscope. So the contents, what is contained in the styrate is used to prove if it is having the sugars, then one can prove that really the sap from phloem tissue is glucose. Therefore, one can conclude that the phloem translocates organic food substances. Here with me, I have the activity, but this activity is in form of the review exercise from what we have discussed. You need to go through that one and try to answer the questions. Now, the first one, you have to state two components of the vascular tissues. Number two, you have to explain any two structural differences between the xylem and the phloem. Number three, you have to state any one functional difference between xylem and the phloem. You have to name any two types of modified cells that are found in the xylem tissue. Now, finally, you have to write an essay, and it's based on the experimental essay. Now you have to describe an experiment that you can conduct to show that phloem translocate organic food substances. So you have to write your answer in essay form, and you have to include the materials, procedure, and the conclusion. Now, the other question here, we have the figure below represents a transverse section through a young stem. Now, this one is the stem, which is labeled A, B, C, C that, D, V that, then E, F. The picture is again adapted from Achievers Senior Secondary Biology, Book 3, page 98. Right, questions on number 6. Question A, you have to suggest whether it is a monocotyledon or a dicotyledon. So you choose an option. But B, you give a reason to support your choice, to support your answer in A above, 6A above. C, you name the parts labeled A, B, D, E, and F. Then finally, you have to state one function of the labeled parts, B, C, and D. So you have to state its functions. Then we have resources. We have the references in Syllable 2017, Excel and Succeed, Biology Book 3. We have Chimocha S 2017, Achievers in Biology Book 3. Now illustrations, design, and graphics. These are by Charlotta and Dave Mumba, who own work in Xulendo. Then photos can be accessed on these links. Then these are the books that we have taken the photos at the end. So basically, this lesson has looked at the structural differences between the xylem and the phloem. It has also looked at the functional differences, structural differences. Their functional differences, how are they different? It has also looked at the activity, the experiment that is conducted to basically prove whether the phloem as really conducts sugar. So once again, I would like to thank you very much for being such a wonderful audience. God bless you, and this marks the end of the lesson. But